Hi, my name is Robert Rosenblatt. I work in the Frank Lab with Dr. Scott Brooks. Today I will be presenting on my research. Pulse-focused ultrasound induces intracellular calcium signaling in TCMK1 cells and the implications for regenerative medicine. So first for a bit of background on this study. So we've seen that pFUS induces stem cell homing, and we know this is through a calcium-dependent signaling pathway. This was figured out by a previous researcher, Rebecca Lorson. So in the first image, you really see the pFUS treatment in, I assume, an animal model to the vessel. And we know after pFUS treatment, you get this calcium increase that leads to NF-kappa B and COX-2, and COX-2 images are on the bottom that leads to chemoattractants and further MSC tropism. But we really want to determine the calcium dynamics that went into this process from the initial pFUS to the NF-kappa B. We know TRPC1 and VGCC are involved, but we don't know much beyond that. Okay, so first to discuss my methods, we used cultured TCMK1 cells and we treated them in either a glass bottom dish or an Eppendorf tube. And I did one of these two experiments on them. I would either load cells with the fluorescent calcium indicator and then live cell image them, which you'll see in traces on the next slide, or I would transfect cells with a GFP reporter, with a GFP NF-kappa B reporter plasmid, and then assay those and read them on a microplate. So here on the left, I'm showing a cartoon I made to really emphasize our process, and I'll take us through a step-by-step version of it. On the right side, you're seeing the trace images from the experiment I mentioned before, and our NF-kappa-B GFP signal graph, bar graph. So as you can see, we added BAPTA here, which is a calcium chelator, and when we added BAPTA, we got no signal from pFUS. This suggested that this is indeed a calcium-dependent process. So here I'm going to show you the first step in the process. I highlighted it. So after FUS, we get activation of the TRPC1 channel, which is a mechanical stretch receptor, which allows entry of sodium and calcium into the cell. And this causes further depolarization of the voltage-gated calcium channels, VGCC, which allows further calcium entry into the cell. We verified this through the use of verapamil and the TRPC1 knockout. Verapamil is a VGCC inhibitor. And what we saw was that we got no NF-kappa B signal after we added either of these treatment conditions. Next, we looked at ER calcium release. As we expected, this was where most of the calcium release came from and is what the big wave in the trace is represented by. So first what we did was we used stapsigargan, which basically knocked out SERCA, which caused the endoplasmic reticulum to empty. And when we used stapsigargan, we saw no pFUS response, suggesting that the endoplasmic reticulum calcium was indeed involved. Then we looked at specific receptors like the ryanodine receptor and IP3R. Ryanodine receptor is caused by calcium-induced calcium release. So this pathway made a lot of sense, and when we knocked out or inhibited the ryanodine receptor, we saw no NF-kappa-B signal. And also when we knocked out the IP3 receptor, we saw no NF-kappa-B signal, which suggests another pathway we hadn't looked into yet. Okay, so to determine the significance of the IP3 receptor, we looked at knocking out phospholipase C or inhibiting phospholipase C, and we also saw no NF-kappa-B signal, suggesting that it was this pathway to a G protein coupled receptor that activated the IP3 receptor. So after doing this, we need to figure out what ligand was attaching to the G protein coupled receptor. First, we thought it could possibly be TNF alpha, and but it ended up not being this because when we did a TNF alpha receptor knockout, we did not see any difference in NF-kappa-B from our pFUS controls. So then we hypothesized that ATP was also released into the extracellular side after pFUS treatment. So to test this, we used ATP gamma S, which is an ATP competitive inhibitor. And when this was used, we also saw no pFUS signal, suggesting this was involved. Finally, we need to figure out what the G protein coupled receptor was here. So we looked at both P2Y and P2X that are that respond to the ATP ligand, and we found that P2Y indeed had no pFUS signal, which was good because P2Y was a G protein coupled receptor, while P2X is an ion channel. So this really fully completed this pathway as to how the IP3 receptor is involved. Finally, we looked at extrusion and store refilling. None of these seem to play significant role. When extrusion was blocked, there was a higher level of intracellular calcium, which is to be expected, and thus our PFUS treatment showed no difference, as the calcium can only be increased to a certain point. And then when ORI1 was knocked down, we saw no difference from PFUS treatment from one sonication. This may be different with multiple sonications, as ORI1 is a store refilling mechanism. So in conclusion, PFUS activation of NFkB requires complex intracellular calcium signaling. This involves TRPC1, VGCC, ER calcium release, and extrusion and store refilling do not seem to be heavily involved. Further, we have to assess the mechanisms of this PFUS-induced ATP release. We didn't fully understand this to this point. And finally, we really need to optimize PFUS parameters for NFkB and COX-2 activation. Any questions?